Hello everybody, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we are going to be talking about the innate immune system, which is so important for our health. We're going to do a quick introduction. We're going to talk about the components and the cells. Thanks so much for being here. So first things first, when it comes to the innate immune system, there are some key things that we need to know about it. Thing number one is it is always present. It is always on, so to speak. And so it right now in all of us is doing what it's supposed to do, helping protect us from potential infections. The other thing is the innate immune system has a cellular and a non-cellular component. We're going to talk about both of those things today. It's not just immune system cells that are helping protect us from infection. Finally, at least in terms of this video, it is mostly non-specific, meaning that the innate immune system generally responds to pathogens in mostly the same way, although we're going to talk about a little bit of a variant from that, but it's for the most part, it's a non-specific entity of our immune response. And we're going to compare and contrast the innate immune system in the next video to the adaptive immune system, which is a little bit different, and then we're going to talk about how the two all work together. All right, so first things first, I want to talk about the non-innate immune system cell components of your immune system, because there's lots of things that help prevent us from getting infections that are not cellular in nature. And so this is basically a list of what we typically have. And so we've got the skin. The skin is by far the best barrier that we have to prevent infection. And in fact, intact skin is very difficult to penetrate, especially when it comes to bacteria and viruses. Yes, there are some protozoans that can sneak their way through hair follicles and things like that, but intact skin is an amazing barrier, and we'll talk about why in a second. The other thing that we have is both mucus and mucous membranes, and that is another physical barrier that we have that separates the inside of the body from the outside of the body, just like the skin does. And that mucus, that is also a physical barrier. And so I like to explain to students, imagine you have this mucus coating in your respiratory system or in the GU. And what's happening there is essentially, remember what we talked about in previous videos. The first thing a pathogen must do is attach to its host in order to cause an infection. Well, that mucus is a physical barrier. And I like to think of it as, imagine you as a human being, we're going to swim across an Olympic-sized swimming pool, but in order to get there, the pool wasn't filled with water. It was filled almost with a jelly. And your job was to swim from one end to the other. That would be really difficult to do. Well, that's the same task that lies ahead of bacteria and viruses when they enter and they get into an area where there are mucus and mucous membranes. They have to get through this mucus layer in order to attach to the actual epithelial cells. But not only that, but contained within the mucus, contained within saliva and other bodily secretions, sweat, tears, there are antimicrobial proteins. And we're going to talk more about what these antimicrobial proteins do, but it would be like going back to that swimming pool example and swimming through that gelatinous material trying to get to the other side, and meanwhile there's a whole bunch of chemicals in there that are trying to attack you as well. And so these antimicrobial proteins, again, they are present right now as we speak in our bodies associated with tears and mucous membranes and saliva. The next thing to consider is pH. Yes, pH, we've talked about that in the past. We have a very low pH, a very acidic pH in the stomach, and then that's also true in the female lower GU, and that pH is designed to destroy proteins. And so again, if you're a pathogen and you're trying to cause an infection and you have to get through the stomach or try to establish yourself in the female lower GU and that pH is acidic, that can be difficult to do. And one thing that often gets overlooked is our normal microbiota or our normal flora. Normal flora are awesome. They're a very important part of protecting us from potential infection. And so again, the point is we have these things, they are functioning in our bodies right now. So let's talk a little bit more about that normal flora and how it protects us. It does a number of things. Thing number one is it takes up binding sites, because we keep saying this, in order for a pathogen to cause an infection, it must attach. And if our normal flora is there and it's taking up all the binding sites, it makes it really difficult for pathogens to attach. Thing number two, it produces antimicrobial chemicals. And so some of the cells that make up our normal flora are producing antimicrobials that can harm pathogens when they try to enter the body. The next thing they do, and something that we don't always appreciate, but it's really important, and that is they prime the immune system. Well, what does that mean? Our immune system will tolerate certain microbes associated with the human body, but the immune system very rarely allows microbes to get into the body, even our normal flora. So if you scratch yourself, there's some type of abrasion, and normal flora enters the body, the immune system essentially has target practice, and it learns how to fight these bacteria as they enter the body. And it's a really important component of a healthy immune system. You need to think of the immune system as kind of like a muscle. If the immune system is not being exercised, if it doesn't have practice in clearing bacteria and viruses, it tends to get weaker over time. And so the normal flora, if they should enter the body, they become target practice for the immune system, and that helps strengthen the immune system over time. And this has been shown in animal studies and human studies numerous times. If you take an animal and when it's born via C-section, and you feed it sterile food, sterile water, sterile environment. These animals don't live as long. They're a lot weaker. Their immune system is absolutely horrible. Why? Because their immune system never got to experience what it's supposed to. In a sense, it atrophied. Just like muscles, they shrink if you don't use them, so will the immune system. Finally, normal flora are super important when it comes to competition, because they can often outcompete potential pathogens. They do an awesome job of colonizing our intestines, our upper respiratory system, our lower GU. And so they're going to usually outcompete the pathogens for food, which means they're also, like I've already said, outcompeting them for space. There are other components of the immune system that are helping protect us all the time, and one of them is known as the complement system. And the complement system has three primary roles. One is opsonization, and that's a fancy word, opsonization. And what that means is these proteins can stick to pathogens, and it makes it easier for immune system cells to recognize those pathogens and then bring them in and destroy them. The other thing that complement does is it's involved in inflammation. It increases the inflammatory response so that immune system cells can get to an area where there's an infection. Perhaps one of the most important things that the complement system does is something called cell lysis. And so the complement system can attach to pathogens, poke holes in those cells, and once there's holes in the cells, the bacteria cells, they can't regulate their osmotic pressure anymore, and they die. And so the three components, again, just remember oil. O, opsonization. I, inflammation. L, cell lysis. And again, these are proteins that are present all the time. Now, they may not be active unless there's an infection, but they're still there. So we talked a little bit about these physical barriers and how important they are. In fact, they're some of the most important components of keeping us safe and protected from pathogens. If we do a thought experiment real fast, imagine you were to swallow something. It could be a piece of food, something undigestible, and it goes into your mouth, into your esophagus, stomach, small, large intestine, and then it exits the body. The question is, did it ever truly enter the body? This is going to sound strange, but the answer really is no. While it got inside, we can't see it anymore. Essentially what we have, our digestive system, is a tube within a tube. And so in order for something to actually enter the body, it has to cross over a physical barrier. That means either the skin or the mucous membranes. And so if that thing that you swallow just passes right through and comes out the other end, it never actually technically entered the body. And this is really important because our body, our immune system, often allows certain microbes, i.e. our normal flora, to colonize the linings, especially the internal linings of our body, the epithelial tissue of our respiratory system, upper respiratory system, digestive system, and GU. What it doesn't do is typically allow for microbes to cross over those physical barriers, breach, in this case, the epithelial tissue, and get into the body. That's the whole priming of the immune system. When microbes do that, even if they're normal flora and are normally tolerated, the immune system will often attack them. Now, there are certain parts of the body where there is more immunotolerance. In other words, there can be situations where microbes do cross over and the immune system doesn't freak out quite as much as it would if the microbes were crossing over in other areas. So the bottom line is, in order for something to get into the body, generally speaking, it has to cross a physical barrier, and that's the skin or the mucous membranes. Now, when it comes to the mucous membranes, it's a little bit easier for those microbes to cross because mucous membranes are designed to, they either secrete things like mucus or digestive enzymes, or they're designed to allow nutrients in, and often mucous membranes are only around a single cell thick. And so it's a little bit easier for pathogens to cross over, and a lot of pathogens have receptors that are specific to epithelial cells that are lining the insides of our body. Skin, on the other hand, is a different story. Skin is watertight. It has lots of tight junctions. The entire upper part of the skin is filled with dead cells. And so even if viruses and bacteria can attach to the skin, there's really no way to, especially for viruses, to gain entry, because even if they attach to dead skin cells, what good is that going to do? There's nothing metabolically active there. And so skin cells are many layers thick. They have those tight junctions. The outside, the most outer layers of skin are dead cells. And so it's really, really difficult for microbes to establish themselves and cross over into the body through intact skin. Now, that doesn't mean microbes can't colonize when skin is damaged, and they often do. There are some protozoans, like I mentioned before, that can sneak through hair follicles and skin pores and things like that. But for the most part, your skin is an awesome physical barrier. Make sure you take care of it. Make sure you're aware of when you have cuts, especially on your hands, because that's a portal of entry for these microbes. Now, I know this is kind of a boring slide with lots of words, but sometimes we just have to show you lots of words. And here is a list of some of the most important antimicrobial proteins or substances. Remember, I talked about this already when I showed you the picture of the person. And we said, hey, there are antimicrobials associated with tears and saliva and mucous membranes. Well, these are some of the really important ones. And so things like things like lysozyme. Lysozyme is hugely important because it degrades peptidoglycan. You can see it's found in so many different parts of the body. And so this is a really useful antimicrobial protein when there are bacteria present, especially those gram-positives where those peptidoglycan layers of the outer membrane are going to be exposed. Peroxidase is another hugely important enzyme. And remember, we've got this ASE, and so that's telling us, oh, this is something that is an enzyme found in saliva and body tissues. Now, this breaks down hydrogen peroxide into reactive oxygen gas. We've actually talked about the opposite in our microbiology labs, where we go from reactive oxygen, which is really bad for genetic material, into hydrogen peroxide, which is also used as a disinfectant because it's kind of dangerous to bacteria. But here what we're doing is there are some cells in our immune system that can reverse, they can take that hydrogen peroxide, which is pretty toxic, make it even more so with these reactive free radicals, these oxygen radicals that absolutely just do a number on viruses and bacteria. Then we have the lactoferrin. And the lactoferrin is an interesting antimicrobial because it sequesters iron. You may say to yourself, well, what good is that? Microbes, just like us, need iron. And if you can take that away, if you can bind that iron for some microbes, that really helps prevent their growth. And then finally, you've got a group of antimicrobials known as the defensins, and these defensins will absolutely wreak havoc on cell membranes of microbes and do some damage there as well. But again, the point is that these antimicrobials are present regardless of whether or not there's an infection, but for sure, they will be in higher concentrations when there is an infection present. There are also an entire group of chemicals known as the cytokines, and these cytokines are designed to basically communicate between immune system cells and even regular body cells, non-immune system cells, with other parts of the body. Really important ones, the chemokines. They're really important for what's known as chemotaxis. We take it for granted if you get an infection that the cells just somehow seem to know where to go. Well, one of the things they can do is follow a chemical scent from these chemokines that are released from other cells. And as that chemical scent gets a little stronger, they know they're in the right place, and pretty soon they start reacting with the pathogens that are in that area. Colony stimulating factors. These are chemicals that tell immune system cells what to become, because as we're going to see in a few minutes, they're all derived from hematopoietic stem cells, and eventually they terminally differentiate into different types of immune system cells. Then we have the interferon. This is a great cytokine that is responsible for preventing viral infections from spreading. We're actually going to talk about this one in more detail here in a couple minutes because it is so important, but it's a really great antiviral component. And in fact, you can actually prescribe, or doctors can prescribe interferon as part of an antiviral protocol. Interleukins are an entire class of cytokines, as these all are really. There are lots of different types of interleukins, and the thing I want you to know about them is the interleukins are kind of like the volume control of the immune response. Some interleukins are pro-inflammatory and make the immune response stronger. There are other interleukins that are anti-inflammatory and basically tell the immune response to chill out, which is really important after an infection is starting to clear, or if there are microbes like normal flora in the body and the immune system is not supposed to be attacking them. And finally, we have tumor necrosis factor. This is an extremely important cytokine that tells cells when to basically go through apoptosis, a programmed cell death, and this is really important for our cytotoxic T cells, our natural killer cells, especially important. This is basically a message that says, hey, you know what, you're infected, you're not doing well, it's time to end this cell life so that the other cells around you can still be happy. All right, so I mentioned I wanted to talk a little bit more about this interferon because it is such an important cytokine. So here we have an example. Basically what's going on is in the upper left-hand corner, we have a cell. And that cell is infected by viruses. So it could be a cold virus, could be influenza, but the cell's infected. Now, unfortunately for this cell, it's going to do two things. One, fortunately for the rest of the cells, it releases this interferon. And this interferon is a message. The message is going out to all the neighboring cells. And so this message is going from the cell in the upper left to the cell on the upper right. And it's saying, hey, cell in the upper right, there's a virus in the neighborhood. Protect yourself. Cell in the upper right says, okay. And as a response, it's going to produce inactive antiviral protein. That's the IAVP. So right now, the cell is just in preparation mode. It's like, hey, I'm not infected yet, but my buddy over there who is told me that there's a virus in the neighborhood. So essentially, I'm going to lock the doors, close the windows, and I'm going to be ready. Unfortunately, for the cell that gave that warning in the upper left-hand corner, it's going to wind up dying. So it's infected by the virus, it bursts open, it lyses, it releases viral particles into the environment around it. So the cell up on the right-hand side, if it encounters a virus like it's doing here now, its reaction to that is going to convert the inactive antiviral protein to active viral protein, antiviral protein. And as a result, you would think with advanced warning, it would do something awesome like beat up the viruses, destroy them. Yeah, it doesn't do that. Instead, what it does is that when that inactive antiviral protein becomes activated, the cell in the upper right undergoes apoptosis, programmed cell death. What happens, though, is you see the cell on the bottom right, that's that cell going through apoptosis. It's not infected by the virus. The virus is not replicating. And so essentially what's happening is those neighboring cells, as they become infected, as viral particles attach, they kill themselves off before the virus has a chance to replicate, and it's a way to slow down the spread of a virus. Sound familiar? I hope so, because in the video when we talked about viruses and bacteria, we said that bacteria have something called the toxin-antitoxin system, and it did pretty much the same thing. If you didn't see that, I'll put it in the upper right-hand corner. It's really interesting. But human cells, something very similar, and that's this interferon. Very, very important. And so sometimes you'll hear when somebody gets infected by a virus and it's a very serious infection, they try to give that person interferon, essentially to do this so the virus spreading slows down. Okay, so we have all of these things that are on, they're present, they're active, regardless of whether or not there is an infection. We also have the cells. We have these cells that are going to help us clear infections or prevent infections from happening. And so the cells that we're going to focus on in this video are innate immune system cells. And then in the next video, we'll talk about adaptive immune system cells. So let's identify those. So what we have here in this picture is we have platelets and erythrocytes. Those are red blood cells and things that carry oxygen. In the middle, you can see what I like to call the phils: basophils, neutrophils, eosinophils. Then we got the monocytes. And from the monocytes, we have the macrophages and dendritic cells. That's where our focus is. Our focus is on the phils, the monocytes, macrophages, and dendritic cells. Those are part of the innate immune system. And then on the right, we have our T cells, our B cells, and our natural killer cells. Those are part of the adaptive immune system, adaptive response, which we will talk about in the next video. All right, so let's start with the phils. You can see they've got some spots. They have spots because they're called granulocytes, and those granules actually hold a lot of cytokines. And so one thing that we could say about all the phils is that one of their primary responsibilities is communication. By releasing cytokines into the environment, they can create a stronger immune response. Now, the neutrophils are the most important. And the most important because they are the most predominant. Somewhere around 50 to 65% of the innate immune system cells are typically going to be neutrophils. The other reason they're important is they are usually the first immune system cell on the scene, partially because they're just so predominant. Their primary role, as I already mentioned, is communication. So when they recognize pathogens are present, they're going to send out that message, those cytokines that say, hey, we've got a pathogen here, time to fight, and they're going to gather up more innate immune system cells to the area. The other thing that it does is undergo a process called phagocytosis. And in phagocytosis, what's happening is that's a fancy word for cell eating. Neutrophils can find pathogens, take them in, and chop them up into pieces. The eosinophils are the next one. Again, with these guys, very small percentages, usually low single digits, 2 to 3% of our total immune system cells, especially innate cells, are going to be eosinophils. So it's a very low percentage. However, what they're really good at is being present when there are parasitic worm infections. However, there have been some studies that have shown when you genetically engineer animals to not produce eosinophils, the parasitic worms are still usually cleared at about the same rate, but the eosinophils tend to be more predominant when there are parasitic worms. The basophils, very, very low numbers, and along with the basophils, you will also hear of mast cells, M-A-S-T, mast cells. These, both of them, like the other phils, will release cytokines. They are both pro-inflammatory. The mast cells in particular release histamine, which is a pro-inflammatory. And so for people who have allergies, sometimes they have to take an antihistamine to neutralize what the mast cells are doing. But again, they are usually in very, very low numbers. Now the monocytes, macrophages, dendritic cells, these guys are pretty awesome. The macrophages, again, this is an introduction to the immune response. If you were to take an actual immunology course, you would learn that there are lots of different types of macrophages, and they have different names depending on where they're at in the body. So the monocytes give rise to macrophages and dendritic cells. Monocytes are the cells that you typically see in circulation. They will differentiate into macrophages and dendritic cells as they migrate into the tissues. Their job is a little bit different from the phils. You can see they don't have as many grains in the cell themselves. And while they can communicate, as all immune system cells can do, it's not their primary role. Macrophages, like the neutrophils, they are involved in phagocytosis, gobbling up the bad guys. Dendritic cells can do the same thing. And so we have three types of cells: neutrophils, macrophages, and dendritic cells that are involved in phagocytosis, the gobbling up of the bad guys, the bacteria and the viruses. However, macrophages and dendritic cells can do something that the neutrophils cannot do. They're known as antigen-presenting cells. And so what they do is they form that link between the innate response that we're talking about now and the adaptive immune response that we're going to be talking about in the next video. Being an antigen-presenting cell, what they can do is they can show off what they found and gobbled up to the adaptive immune system. Now, the dendritic cell can take it a step further. We're going to talk more about that in the next video. But one other thing that the dendritic cell is really important is you can see it's got those little arms. It almost looks like an octopus. It can stick out those little arms in between epithelial cells and actually sample the lumen of the intestines and the respiratory system and the lower GU. And so it can actually detect antigen that's technically outside the body because it's still on the lumen of the intestine before it gets in. And so it can give a little bit of advanced notice there. Now, remember when I said the innate immune system is mostly nonspecific, but I had a caveat there. I said later in the video, we're going to talk about something that's, well, not quite nonspecific. That's these guys. These are toll-like receptors. It's an interesting concept, toll-like receptors. The word toll in German means weird or kind of, wow, that's crazy. And it's kind of like a reaction, a surprise. What happened was back in the 1980s, scientists were studying fruit flies and they were messing with their genetics. And they found out that as they were messing with certain proteins, that fruit flies would do weird things. They would develop legs on their head, the wings would be backwards, and that was their expression, toll. I'm like, wow, that's really something. Well, years later, scientists were looking at the immune system, and they discovered what looked like the same proteins that they were investigating in these fruit flies in human beings, so they called them toll-like. What they were doing, their responsibility in the immune system is they are responsible for recognizing certain conserved regions of bacteria and viruses. And so while a lot of times we say the innate immune system is nonspecific, that's not totally true because we take it for granted that all the cells we just talked about, our neutrophils, macrophages, dendritic cells, well, they recognize the bad guys. They recognize the pathogens. Well, how do they do that? They don't have a brain. They use these toll-like receptors, and they're specific for certain areas of bacteria and viruses. And so there are certain toll-like receptors that recognize peptidoglycan, LPS, capsid proteins, bacteria flagella, double-stranded RNA. And so when they recognize these things, that kind of tells the immune system what it's dealing with. If you've got a toll-like receptor that recognizes peptidoglycan, it's probably a gram-positive microbe. If you have a toll-like receptor that's recognizing LPS, it's probably a gram-negative. If you've got a toll-like receptor that's recognizing double-stranded RNA, that's probably a viral infection. And so based on that, the pathway that the immune system takes can be a little bit different. Bottom line is when it comes to toll-like receptors, this is a way for microbes to actually determine what is a pathogen and what isn't. It's how it recognizes those pathogens. Immune system cells also rely heavily on what are known as adhesion molecules. And these adhesion molecules are basically kind of like the brakes. The adhesion molecules are often produced in endothelial tissue. And as the immune system cells are in circulation, they catch these adhesion molecules, and that allows them to stop where there is the site of infection, get through the capillary, and eventually into the tissue where the bacteria or the virus is. And so let's kind of put this all together. We've got this inflammatory response, and for right now, we're really focused on the innate immune system. And there are certain signs of inflammation that many of you have heard of before, and you should definitely know if you're talking about the immune system. And those cardinal signs of inflammation are often redness, swelling, pain, potential loss of function. And so why is that happening? Well, the reason that's happening is because of the innate immune response. And so here we've got an example. There's a splinter. There's some kind of a breach of a physical barrier. In this case, it's the skin. Immune system cells, in particular neutrophils, are often going to be the first on the scene. Macrophages are going to be right there as well. And so if I were to pick them in order, neutrophils are right there. The macrophages are probably going to be the very next cell that is there. They're going to be sending out pro-inflammatory cytokines. Now, this is going to do a couple things. It is going to dilate the vessels that are headed to the wound, and at the same time constrict the vessels heading away from the wound. The whole point is try to keep whatever's entered the body, whatever's crossed a physical barrier at that location. We don't want it spreading all around. And so when you have that influx of fluid because of the dilation of the vessels on the way to the wound to get the immune system cells there, and you have constriction on the way out, you're going to get the swelling, pain, redness. Another one is heat and loss of function. And that heat one is really important because that heat, that increase in temperature, is partially responsible for converting that complement protein from the inactive form to the active form. And so then the question becomes, when there's inflammation that is due to injury, should you actually reduce the inflammation? There are some who say when you do that, you're actually extending and making it more difficult for the immune system to do its job and potentially extending the response that's going to happen. Others will say, and possibly and rightfully so, if the inflammation is chronic, well, that can do a lot of damage too, because some of the things that we've talked about so far, if they are chronically high in high concentrations, can cause damage. So for instance, this complement system that we said causes cell lysis, if that remains active for too long, eventually that complement system is not just poking holes in our own cells or in bacterial cells, it's poking holes in our own cells as well. And that can be a huge problem. Some of these pro-inflammatory cytokines can be a problem. As we look later on down the road at the adaptive immune response, we get things like antibody antigen complexes that are binding that just stay in that area, and more inflammation happens, and that can cause damage over time. However, again, the point is, inflammation just due to injury or something like this, a splinter, that can be a good thing. That's the immune system trying to do its job. The pain that you're feeling is telling you, hey, this part of the body is injured right now. Please don't make it any worse. Chronic inflammation is something you really should consult your medical professional about, your primary healthcare physician. One of the things that gets overlooked a lot are these guys here, and that's the platelets. And it turns out they are an important part of the immune response. Many of us know that platelets are responsible for blood clotting. And so when you do injure yourself, like in the previous slide, we just talked about splinters or some kind of a breach, platelets are there to make sure that we get that separation from the outside of the body and the inside of the body. That's great. Turns out platelets are more important to the immune response than even that. They, like immune system cells, may have toll-like receptors, which means that they can recognize when pathogens are present, which is awesome because that greatly enhances our surveillance capacity in our body. Not only that, platelets have been shown to release pro-inflammatory cytokines. So not only can they recognize, and it kind of makes sense, if you can recognize a pathogen, hopefully you can do something about it, at least alert the immune system that something is present. Turns out they can. And so this is a really interesting component. Platelets not only involved in blood clotting, have toll-like receptors, and can release pro-inflammatory cytokines. Last thing I want to talk about before we leave this concept of the innate immune system, that is a fever. When your body is increasing the temperature, just like we talked about with inflammation, when it comes to fever, it is your body's attempt to make the infectious agent uncomfortable. Take it out of its comfort zone. We know this from a previous video. We talked about optimal temperature and pH and osmotic pressure for enzymes. Well, when your body increases the temperature, some of the microbes that infect human beings, they don't do so well because now they're outside of those optimal conditions. Now, obviously, if a fever gets too high, that can have an impact on us as well. And so again, the question becomes, should you reduce a fever or should you just let the fever happen? Again, you need to talk to your primary health care provider about that, but a fever is designed to make microbes uncomfortable and to try to slow their growth. All right, everyone, so that is a brief overview of the innate immune response. In the next video, we're going to be talking about the adaptive response, and then we're going to be tying these two things together so that we understand how they both work together in a really coherent way. As always, thank you so much for being here. Have a good week, everyone.